We are honored to have Dr. Ethan Russo presenting for us in our first public seminar series. The topic today will be exploring cannabis science and the endocannabinoid system. The agenda for today is this welcome. I'm going to give a brief CRI overview. I will introduce Dr. Ethan Russo, who will give his talk, and then we will do a question and answer afterwards. All right. So for those of you for those of you joining us for the first time, I'm going to give you a bit about CRI. We are funded by the Illinois Department of Human Services with supplemental funding from the Cannabis Research Oversight Office. Our mission is to foster public understanding and change the stigma associated with cannabis. We do this through rigorous research and programs that are community-centered. Our focus is to promote equity, provide opportunity, inform policy, and through a collaborative, interdisciplinary approach, turn science into impact. Our goal is to be the leading trusted institute for community-informed cannabis research, and as part of our daily work, advance equity and sustainability. So this is our second year. Our first year started July 1, 2024. We had a number of milestones that we hit. One of which we're very proud of was we had our initial experiential learning cohort, our foray into trying to figure out what we could offer and how we would offer hands-on lab experience. Our year two focus. Let me finish by saying we had a number of outstanding students, some of whom are actually going to be co-authors on papers and potentially first authors on some of those papers. So our year two focus will be continued community engagement, expanding our educational programs through additional internships and supporting community college-based cannabis research programs, community-guided research for which there will within the state of Illinois potentially be a request for proposals for some seed funding. And we also have our internal and collaborative research, right? So all of which generates information that will be amplified and disseminated without bias, meaning that no matter what the data says, that's what we will share, right? So we are not looking to use our platform to convince anybody of anything. We want to just provide unbiased, rigorous data that will inform policy and help people understand the plant better. So I mentioned our internal and our collaborative research. This slide, so there's a little inaccuracy here, and it's my fault. So collaborative library research should probably actually be research where people came to us and asked us to participate. We have we had two projects that we were involved in or will be involved in if the funding comes through. Cannabinoids for pain mitigation in endometriosis and sickle cell disease, and cannabinoids as inhibitors to sickle cell disease-induced retinopathy, which is a loss of vision. So we were approached by UIC, which is the University of Illinois Chicago, for those of you not within state, UIC researchers to participate in that. On the other side, our current in-house laboratory research is the research that's started by us in-house, but we also do that collaboratively as well. So the identifying compounds in cannabis and hemp that are effective against cancers, that's what we're doing that research with Northern Illinois University. And the other two projects, understanding relationship between terpene enzymes and the products that are made, and developing breeding markers for terpene genes, are all being done in-house with our in-house staff. So at this point, I would like to introduce Dr. Ethan Russo. Ethan is an MD, board-certified neurologist and psychopharmacologist. He's the founder and CEO of Credo Science, a company devoted to making cannabis safer and better. He has held leadership positions in major research organizations and the pharmaceutical industry related to cannabis and the endocannabinoid system. He is the author or editor of 7 books and has published over 70 peer-reviewed scientific journal articles. Please join me in welcoming Ethan and for his presentation, Exploring Cannabis Science and the Endocannabinoid System. Ethan, off to you.
Thank you, Reggie. I am just going to set up the slideshow and get this moved. So, welcome everyone. It's a pleasure to initiate this series of presentations. Today we'll be exploring cannabis and the endocannabinoid system science. So first, what is the endocannabinoid system, henceforth the ECS? So the endocannabinoid system is the major homeostatic regulator of human physiology in virtually all its facets. Let's break that down. So, physiology is how our body works, and wherever you look in the body, whether it be digestion, uh, nervous system function, even regulation of bone, its breakdown and rebuilding, all that is under the direct control of the endocannabinoid system. So if there's too much activity in the system, it brings it back down into a normal range. If there's an underactivity, it brings it back up into the normal range. And we'll talk about how that works. So how did this come about? Our knowledge of the endocannabinoid system goes back a little over 35 years, but it all started with the plant Cannabis sativa, cultivated cannabis. Um, we see the plant here, and it has glandular trichomes, these little things like golf balls on stalks. That's where the cannabinoids are made. The most notorious among them is THC, delta 9 tetrahydrocannabinol. And it turns out that its function is closely related to natural chemicals within our bodies called the endogenous cannabinoids or endocannabinoids. And they work on cannabinoid receptors. So the two best known are anandamide and 2-arachidonoglycerol, and they were discovered in the early 90s. As it turns out, THC as well as the endocannabinoids bind to this receptor in the brain, um, and we'll show how that works. Now, the cannabis plant was not put on God's green earth to make us high. Rather, this system goes back in time, a great deal of time, about 600 million years evolutionarily. And the endocannabinoid system is found in all vertebrates, animals with a backbone, or chordates, the more primitive form. So it's 600 million years old. Just for comparison, the cannabis plant itself is 30 million years old, and the human species is less than 1 million years old. Interestingly, insects don't have an endocannabinoid system, and I like to say that's why they don't have a sense of humor. The This is a study from the dawn of the endocannabinoid era. So they used something called CP 55940, which is a very high potency CB1 stimulator or agonist. They use this to map CB1 receptor in various species in the brain. And we'll just use one example here. Where the red arrow is, you see the human representation of the CB1 receptor in the cerebellum that is involved with balance and coordination, among other things. And you'll see that it's much more dense in the dog. Interestingly, 100 years ago, when cannabis preparations were part of the pharmacopoeia and were available as prescription items, they used dogs to test the potency of the cannabis because they didn't have biochemical assays at that time. Also important are changes in the density of the receptors, endocannabinoid receptors at different stages in human development. ECS is critically involved in brain development and architecture, what goes where when. So in terms of embryology, it's a key function again. Interestingly, the density of the CB1 receptor, where THC and the endocannabinoids work, is much greater in the fetus and in children. and that may be an explanation for why kids seem to be naturally stoned. As it turns out, the three kinds of cannabinoids. We first knew about phytocannabinoids, so those derived from plants, such as cannabis. And this term has been applied to structures with 21 carbons found in cannabis, especially THC and CBD, cannabidiol. But as it turns out, they're elsewhere in nature, as we'll see subsequently. We've also introduced the endocannabinoids. So these are natural endogenous compounds that bind those same receptors. Anandamide was the first discovered, and its name was for Ananda, which is Sanskrit for bliss. Professor DiMarzo characterized its functions as relax, eat, sleep, forget, and protect, but we could add 30 other functions and it would still be accurate. And there are also synthetic cannabinoids. One example is agilamic acid. So these would be synthetic substances that also affect the cannabinoid receptors. The two main cannabinoid receptors are oddly named CB1 and CB2. So CB1 was first. It's in the central nervous system, the peripheral nervous system. It's also in the gonads, heavily represented in female anatomy. So among many other functions, this modulates pain, movement, emotion, whether you need to vomit or not, whether you'll have a seizure or not. A few years later, CB2 was discovered. This is mainly out in the periphery. It only appears in the brain under conditions of damage or stress. So it is an immunomodulatory receptor, also involved in inflammation and pain. Now, whereas the structure of CB1 is similar in most species, it's very heterogeneous in different species, CB2 that is. And in fact, there are variations of CB2 that may occur within species. So if you see a study that was done with rat CB2, it is really questionable whether that applies to humans or not. And this is a reason that frequently studies are done with humanized CB2. Let's define the activity of the receptors. A word you'll hear a lot is agonist. So these are substances that stimulate the receptor, and they can be weak, partial, or full. Interestingly, THC is a partial agonist at CB1 and CB2. So that means that it operates with some subtlety. And there are two kinds of antagonists. Neutral antagonist is one that will take its residence on the receptor, but it doesn't alter the activity overall. An example there is what's called tetrahydrocannabivarin or THCV. And there also can be inverse agonists, which is an unwieldy name, but it means that it actually decreases the overall activity of the endocannabinoid system, lowering what we call endocannabinoid tone. So an example there is SR141716A, known as rimonabant, which was briefly on the market in Europe as a weight loss drug. But because it lowered endocannabinoid tone, it was responsible for a great deal of depression, anxiety, and generated a few seizures and even a case de novo of multiple sclerosis. So there are two different kinds of receptors. The orthostatic, which is the main receptor, or the allosteric, that means another site that affects the binding of the receptor. So those are all allosteric modulators. These also come in two flavors. They can be negative allosteric modulators. Actually, cannabidiol operates that way at the CB1 receptor. And there can be positive allosteric modulators. One example is the synthetic ZCZ11. There may be natural examples as well. The search is on. Now, the way that this cannabinoid receptors work in the brain on the neurons is very complex. we usually think of neurotransmitters as going in one direction, from the presynaptic neuron to the postsynaptic neuron. In fact, CB1 works oppositely. It is produced on demand and they travel backwards or in a retrograde fashion. So whether it's THC or anandamide, when it stimulates CB1, what this will do is inhibit the release of neurotransmitters. So if this is a stimulatory neurotransmitter like glutamate, the effect of CB1 stimulation is going to be to reduce that activity. If, on the other hand, this is a GABA receptor, which is an inhibitory receptor, if you inhibit the inhibition, it acts in a stimulatory fashion. And there are also regulatory enzymes for all this. As we'll see later, in the body, it's not just 2-AG and anandamide, but there are actually hundreds of related endogenous cannabinoids, some of which are inactive but may work on their own, but together they produce what's called an entourage effect. CB1 expression in the brain is variable. There is a density of CB to nociception or pain. Also in the cerebellum, as we've seen, the limbic system centers for emotion, basal ganglia that affect movement, and in reward pathways. There are less CB1 receptors in the brainstem, but there are in certain areas, the substantia nigra and periaqueductal gray. The latter is important for pain thresholds, but not lower down in the brainstem. So breathing is controlled in the medulla, the lowest level of the brain. And because there are hardly any CB1 receptors there, there is no dose of THC or cannabinoids that will produce apnea, stopping breathing, as occurs with the opioids. We've seen this slide before, but to reiterate, the endocannabinoid system is everywhere. It's in the brain, it's in the spinal cord, and it's very much involved in various aspects of pain transmission. It's important to understand that CB1 is the most abundant G protein coupled receptor in the brain. There are more receptors for CB1 than there are for all the neurotransmitters combined. It's also in the gut, where it modulates propulsion, how fast things are moving, and secretion, how much liquid is there. And it's out in the periphery too, even regulating things like whether you itch or not. Now on to CB2. As mentioned, this is mainly a peripheral immunomodulatory receptor. It has important functions in inflammation and pain. It also has an antifibrotic effect. So agents that stimulate CB2 would be helpful in liver fibrosis and similar conditions. A very elegant paper from years ago, Professor Mishulam and Professor Pacher, showed the effects of CB2 in all these different organs of the body and indicated that there was a protective effect here that was distinct from what would be involved in treating infections, but there's a widespread importance in all these peripheral organs. But it also is in the skin. We have CB1 and CB2. Interestingly, cannabidiol works on the skin. It's working on another receptor, which is considered part of the broad endocannabinoid system called TRPV4. And it stimulates that, and this actually reduces the amount of sebum, the waxy substance in the skin that is the feedstock for acne bacteria. I appreciate this graphic that we requisitioned some years ago because it looks like a heartless person with no backbone to speak of. But in fact, the endocannabinoid system is in the heart and bone as well. Interestingly, cannabidiol stimulates bone fracture healing, and if the laws change, it may be that CBD is going to be prescribed to athletes in the emergency department when they have fractures. Now, there are additional receptors, particularly TRPV1, which is called an ionotropic cannabinoid receptor. It was discovered because that's where capsaicin, the hot ingredient in chili peppers, works. And CBD works there as well. And this is how it works on the receptor. Um, it sort of mirrors where CB1 is. It's in the spinal cord, in brain areas. It's also heavily involved in emotion, something you wouldn't think about in relation to chili peppers, but capsaicin is not absorbed into the body. So that this receptor is involved in neuropathic and inflammatory pain, itch, the tone of the blood vessels, excitotoxicity in the brain. It can have effects on incontinence, cancer, all these different things. So this is a very important system. Um, Interestingly, agents that stimulate the TRPV1 receptor will also sort of turn things off. They produce a desensitization. And we won't go through all of this, but it's important because instead of an antagonist, which has been tried on this system and have not worked out clinically, you may have to fight fire with fire. And so capsaicin is an example of a stimulator of TRPV1 that's pungent. That's why it burns. But there are also non-pungent examples like cannabidiol. So in terms of therapy, it would be ideal to have an agent that activates the TRPV1 receptor, but not producing pain when it's first applied. So would be less pungent, but produce the desensitization. And CBD may fit that bill. In terms of its potency, cannabidiol is quite analogous to capsaicin in binding. And it is capable of desensitizing the TRPV1 receptor and could be useful to turn down the heat and pain. So that would apply to all these different conditions that we see here. Many forms of neuropathic pain, like complex regional pain syndrome, especially skin burns, which continue to burn long after the fire is gone. Things like irritable bladder, interstitial cystitis and prostatitis, chronic pelvic pain, fibromyalgia affecting the muscles and soft tissues. Also, things like inflammatory bowel diseases and irritable bowel syndrome, pancreatic pain, which is extremely difficult to control, but also things like itch and allergies of the skin. It could be useful in anxiety treatment. And it likely would have a role in treatment of cannabinoid hyperemesis syndrome. We're not going to cover that today, but you can see papers on that at ethanrusso.org at the library tab. We also should address how the endocannabinoids are made and broken down. Anandamide was first, again a partial agonist at CB1 and CB2. It is made from a thing called N-arachidonyl phosphatidyl ethanolamine, easy for me to say. And it's broken down by several different enzymes, but especially fatty acid amide hydrolase, which has been a target for a lot of pharmaceutical development. Keep in mind that cannabidiol and cannabigerol both act on that. And 2-arachidonoglycerol, it seems to be a full agonist at CB1. It's actually more of it, but these two have different roles. It's made from diacylglycerol lipase and is broken down by various different lipase enzymes, but especially one called monoacylglycerol lipase, or MAGL. Now, one of the questions that has come up is, what happens if you have too much endocannabinoid system activity? Well, we see a lot of this now because of the obesity epidemic. So we see this with the metabolic syndrome and type 2 diabetes. As mentioned, the drug rimonabant was an inverse agonist was used to treat this, but the adverse events were unacceptable and it was taken off the market. There's been a lot of research with neutral antagonists like THCV or uh, NAMs, um, the negative allosteric modulators like cannabidiol that don't have those side effects. We also can have underactive endocannabinoid function. Now, this is a theory I developed, first published in 2001, um, and I called it clinical endocannabinoid deficiency. The theory was all humans have an underlying endocannabinoid tone. That is a reflection of how much anandamide and 2-arachidonoglycerol you have, what the state of their production is, how they're being broken down, and the relative abundance and state of the cannabinoid receptors. The theory was that there would be conditions, whether you were born this way or developed it, that endocannabinoid tone is too low and that you have problems because of it. Now, I developed this theory as a neurologist because we knew that so many diseases are due to deficiencies of neurotransmitters. Uh, in Alzheimer's disease, there's not enough acetylcholine, the memory molecule. In Parkinson's disease, there's not enough dopamine. And in depression, there may be alterations in serotonin function. So, why would there not be people who suffered endocannabinoid deficiency? In the first paper that I wrote on this in 2004, my prime candidates were migraine, fibromyalgia, and irritable bowel syndrome, but you'll see that there's also a longer list here. What these have in common is they're what's called diagnoses of exclusion. There are no tests for them. If you look at the tissues, they look okay, and yet people have bad pain associated with these. In the case of migraine, even light is too bright and noise is too loud. Fibromyalgia can produce pain throughout the body. Irritable bowel is associated with gut that hurts. Um, and these three conditions are comorbid. That means they tend to occur in the same individuals. Um, and interestingly, cannabis is used to treat all of them. And often the conventional drugs used to treat them are not that effective. Now let's turn to the plant, Cannabis sativa, cultivated cannabis. I like to call this the archetypal desert island plant because if you had to choose one thing, if you were on the proverbial desert island, it should be this plant because it can provide food from the seed, fuel from the seed oil, fiber to clothe yourself from the stalks, and then, of course, its many therapeutic effects, mainly from the unfertilized female flowering tops. This is an interesting plant because it has sexes on different uh, plants called dioecious, and it's an annual. It can grow 6 meters in 1 year. So it's a really amazing plant. Now, there are different types chemically. What we like to call chemovars. There are no strains in cannabis. Strain is a term that applies to bacteria and viruses. Type 1 is most familiar. These are high THC chemovars, chemical varieties. Type 2 is mixed THC and CBD. Type 3, CBD predominant. And then newer types, cannabigerol predominant chemovars, type 4. Type 5, plants with no cannabinoids at all. And type 6, tetrahydrocannabivarin predominant. This is from an old paper looking at the plant, and actually, there are many different products from different parts of the plant. Again, we look for the cannabinoids as concentrated in the glandular trichomes. We have the seeds, which are excellent in terms of their protein and beneficial fat content with essential fatty acids. And we have the roots, which have no cannabinoids but contain alkaloids, something that was looked for in the flower for decades, and there are none. Um, These are 12 of the most common of the plant-based cannabinoids in cannabis, the ones where we know the most about their biochemistry and pharmacology. And we'll just deal with a few. But first, this is how they're made. Usually, it's a combination of olivotolic acid and geranyl pyrophosphate. These produce first something called cannabigerolic acid. All the cannabinoids in the plant are produced in acid forms, and it's only after they're subjected to heat, light, or age that decarboxylation, a loss of the CO2 group, occurs, and that will lead eventually to THC, CBD, and the others. Geranyl pyrophosphate can also combine with another agent, divernic acid. You'll notice that this has 3 carbon side chain instead of 5, and this produces a whole series of other cannabinoids, which now number about 150. But these are called the Varin series of propyl cannabinoids. Interestingly, geranyl pyrophosphate is also the parent compound to the terpenoids in cannabis that we will also discuss. Back in 1998, Professors Meshulam and Ben Shabbat described an entourage effect, a combination of the main players, anandamide and 2-AG, along with related compounds that produce synergy between them. The next year, they published this study and said, this type of synergism may play a role in the widely held but not experimentally based view that in some cases plants are better drugs than the natural products isolated from them. So they said that that was theoretical in 1999, but there's been 26 years since, and a lot of evidence that this prescient analysis was in fact accurate. So, as I've said, the endocannabinoid system is a lot older than cannabis is. So, why does the plant go to the trouble to make this stuff? Well, we know that cannabinoid production is greatest in bright light, so it may offer some protection against ultraviolet light damage. Some of the agents in cannabis are antifungal, and these trichomes have a form of mechanical insect trap. But additionally, the acid cannabinoids are also insecticidal. So we have a double whammy for the insect that strays too far. Um, and also, the acids will have the effect of auto pruning the plant as it matures. So if we prevent fertilization, what was a very densely foliated plant becomes very lanky as it's trying to get fertilized, which is something that's prevented by humans cultivating the plant to increase the cannabinoid content. Um, some detail about the cannabinoids. THC was ultimately identified in 1964. As mentioned, it's a weak agonist at the cannabinoid receptors. It has effects as an analgesic, painkiller, and itch. Uh, Bronchodilator, it opens up the lungs. Uh, there's actually a patent on the next one, a neuroprotective antioxidant effect that should help prevent brain damage from a variety of insults. THC has 20 times the anti-inflammatory power of aspirin and twice that of hydrocortisone. It's a muscle relaxant. That's the basis of its approval in Sativex as a spasticity treatment in multiple sclerosis in 30 countries outside the US. Since 1985, THC has been used as a synthetic to prevent vomiting associated with chemotherapy, and in 1992 it was approved for AIDS wasting syndrome. This, of course, is the primary psychoactive component. It also has a role in reducing beta amyloid, one of the abnormal protein accumulations in Alzheimer's disease. Now, cannabidiol has been received less attention until the last 20 years, but actually is a more versatile agent, although much less potent. This was isolated in 1940, but in 1963, Professors Meshulam and Schvo figured out exactly what it looked like. As mentioned, it's not working directly on CB1, but is a negative allosteric modulator. It makes it a little harder for THC to bind and can thereby reduce the side effect profile. It also is a neuroprotective antioxidant. It helps reduce brain damage associated with glutamate excess, as in head injury, and it's stronger as an antioxidant than vitamin C or vitamin E. As mentioned, it works on the TRPV1 receptor as well. It also will increase the amount of anandamide over time by inhibiting its uptake and its breakdown. So it can, over time, increase endocannabinoid tone. Um, it also is an anticonvulsant. An approved drug in the US is Epidiolex, which is 98% pure CBD, effective in seizure syndromes such as Dravet and Lennox-Gastaut syndromes. In very high doses, hundreds of milligrams, it has an anti-anxiety effect. It will affect cancer cells. It's cytotoxic, like chemotherapy, at high doses. On almost all the cannabinoids have this function, and interestingly, some work better in different types of cells. It also is an antagonist to what's called tumor necrosis factor alpha. This is a very important in autoimmune diseases like rheumatoid arthritis and multiple sclerosis. It has this very powerful anti-inflammatory effect without affecting the COX enzymes. So almost all the standard anti-inflammatories work as COX inhibitors, and this produces the side effects of ulcers, gastrointestinal bleeding, strokes, etc., that cannabidiol doesn't do. In 2005, it was unknown why cannabidiol had anti-anxiety effects. But with my colleagues at the University of Montana, we hypothesized that it was actually working on serotonin receptors, and that turned out to be true. And it also is responsible for possibly some of the anticonvulsant effects, its benefit in treating strokes, and also as an agent to help prevent vomiting. Again, working on adenosine receptors, this can also be helpful in various inflammatory and chronic pain states. Some years ago, it was shown that this might help prevent slow virus infections, prion accumulation. Now, there are a lot of misconceptions about cannabidiol. It is a very versatile agent, but it's not potent. Often we need very large amounts if it is a pure compound. It works better with the other agents in cannabis. So a tiny amount isn't enough. More is better. This is commonly mischaracterized, even in published journal articles, as not being psychoactive. That's nonsense because it is an anti-anxiety agent, and in very high doses, it has an antipsychotic effect as well. So what we should say is that it's not intoxicating in the way that THC is. You also hear that it's sedating. It's not. At low and moderate doses, it actually has a stimulating effect. And some people in California use cannabidiol in the morning instead of coffee. Another myth is that it turns to THC in the body. It is true that you can artificially make THC from cannabidiol with strong acids, but there's no mechanism in the body for that to occur. This is a sort of schematic from an old article going from the field to pure CBD crystals. You've already seen the metabolic pathway. And as mentioned, this is metabolized to something called 7-hydroxycannabidiol. It is not turned into THC in the body. Cannabigerol is a very interesting agent. It is sort of the mother of all cannabinoids. This was also discovered by Meshulam et al. They called this the missing link in the plant synthesis of cannabinoid constituents. Initially, they didn't see a lot of changes in test animals, but over time, we noticed all these other effects. It's a GABA uptake inhibitor. This could be responsible for its ability to have some anti-anxiety effects and reduce spasticity, muscle tightness. It's antifungal. Some of its effects on pain might be related to the alpha-2 adrenal receptors. Just recently, Guvanlu et al. Showed that this works on a peripheral channel called the NaV 1.8. And this portends to be useful in pain treatment without any ability to cause intoxication. Like THC, it lowers intraocular pressure. Like CBD, it also works on various cancers, especially breast cancer and as well prostate cancer, because it works on something called the TRPM8 receptor. May have application in reducing buildup of excess skin and psoriasis. And it's a powerful agent against gram-positive bacteria like multiply resistant Staph aureus. And this is part of an agent in development now with cannabichromene and silver as a broad spectrum antibiotic working also on gram negatives and fungal agents. Let's see. This also inhibits anandamide uptake, so it could increase endocannabinoid tone. It also works on the nuclear receptor, PPAR gamma, and this may have great implications for cancer, metabolic syndrome, etc. It has a particular activity on glioblastoma multiforme, the worst kind of brain tumor. Just last year, with Kerry Cutler, we published a study showing that CBG in a moderate dose reduced anxiety acutely in a randomized controlled trial and actually improved verbal memory, showing no impairment. Now let's turn to terpenoids. These are what give cannabis its smell. Again, they're not there to make the odor pleasant for humans, although we can breed for that. But a lot of these have insect repellent activity or an insecticidal. And different ones are produced at different levels in the plant. So there are a lot more bitter ones at the bottom that keep Bambi from grazing on the plant. And it has strong antibiotic properties. This is again from an old paper, Taming THC. And as you'll see, certain terpenoids from cannabis, they're not unique to cannabis. We see these in more common plants, so you get an idea of what these smells are like. So some of these are painkillers, many are anti-inflammatory, many have effects on mood, and they also can modulate THC effects in a synergistic fashion. We'll just discuss one today, which is beta-caryophyllene. This has a distinction of not just being a terpenoid, but additionally, it's got to be considered a cannabinoid because it is a high potency selective agent at the CB2 receptor. So it was known early on that this had a very strong anti-inflammatory effect, and balsams from South America have been used long to treat wounds. So this is as strong as phenylbutazone on anti-inflammatory, so strong it's not used in humans but in horses. Unlike the anti-inflammatories, though, this actually protects the stomach from damage. It again is a full agonist at the CB2 receptor, and it seemingly has a role in treating addiction. So this is an agent you'll be hearing more about. Synergy. This is the idea of 2 plus 2 normally equals 4. Can you get increased bang for the buck with different combinations of ingredients? an early demonstration of this was from a study of Sativex for treatment of cancer pain. Um, in this study, there was a comparison of a THC predominant extract against placebo and compared against an extract high in THC and high in CBD. And it turns out that the combination was better. Here we see it graphically. This is placebo. This is the high THC extract. No significant difference. But in terms of reducing pain by 30% or more on cancer pain, there was a clear improvement with the combination of the high THC and high CBD extract. This would be an indication of synergy. Another example would be in primary treatment of cancer, in this instance, breast cancer. So in the study, they compared pure THC to an equivalent amount of THC in an extract. And if you look carefully on the left, there are statistically significant differences. So what can we attribute that to? Well, small amounts of CBG and tetrahydrocannabinolic acid seem to make the difference. So this is something, again, that's being actively investigated. Now, this is too long to read, but I had mentioned previously that CBD as an isolate, a pure compound, is not very potent. The amount of CBD that's necessary to treat these severe seizure syndromes is very high. It can be in an adult 2,000, 2,500 milligrams a day. But in this study that compared Epidiolex, pure CBD, to other forms of CBD where they were from extracts with other components, what was required to seemingly produce the same level of control was about 20% of the CBD concentration that was necessary as an isolate. I believe that this is due to other components because there are various cannabinoids and terpenoids in cannabis that also have anticonvulsant effects. This would be an example of a chemovar like that. So we see straight off, this has cannabidiol. It has almost as much cannabidivarin. Cannabidivarin works on other kinds of seizures in a distinct manner from CBD. It also has high amounts of caryophyllene and linalool. So in fact, all of these agents, CBD, CBDV, THC, linalool, have anticonvulsant properties, and maybe the caryophyllene works as well as an anti-inflammatory. This is from an older paper, levels of evidence for cannabis-based medicine. So as we've mentioned, THC is approved for treatment of chemotherapy-induced nausea and vomiting. CBD is approved for intractable seizures in Dravet syndrome, Lennox-Gastaut syndrome, and in tuberous sclerosis seizures. Multiple sclerosis spasticity symptoms with Sativex approved in 30 countries, and there's a great deal of evidence of benefit on adult chronic pain. There are various studies for and against, but moderate evidence of efficacy in treating sleep, fibromyalgia, things of this sort. There have been benefits on symptoms of dementia, particularly agitation, benefits on glaucoma, intraocular pressure treatment, etc. And then a lot of other things being tried, as you'll see on the list. Now we're going to turn a little bit to the effects of diet, because this affects the endocannabinoid system. In our gut, we have 100 trillion microorganisms, and there are more of them than there are of us. And so their genome is very important to how things function. And this will also help decide whether you're going to gain more weight than you want as well. So the microbiota in the gut can affect your mood. It has a role in autism, depression, on recovery from strokes, and also in schizophrenia. Um, the effects are mediated through the autonomic nervous system, the enteric nervous system, and the immune system. This is based on bacterial metabolites. Most of the serotonin that's produced comes from your gut bacteria. Certainly that there are effects on the endocannabinoid system. We have a lot more autoimmune disorders in our population now than 100 years ago. But we've had massive global dietary shifts. So we're seeing an epidemic of obesity, inflammatory bowel disease, other autoimmune disorders, allergies, diabetes, cancers, depression, and neurodegenerative disorders. And it's not just because people are living longer. It can be measured and showed that modern society is more afflicted with these problems, and diet is a likely culprit. Part of what we can do is increase the diversity of our gut bacteria. Part of that is by feeding them properly with what are called probiotics. Um, these may help control the PPAR gamma receptor, which is the master regulator of adipogenesis, that's fat accumulation, also affecting the tumor necrosis factor alpha and inflammation. These pre probiotics are things like yogurt and the lactobacilli. Prebiotics are the feedstock for the probiotic bacteria in the gut. So these also reduce fat accumulation and stabilize the gut barrier. They also affect levels of CB2, and that's correlated to concentrations of the lactobacilli and negatively with Clostridium, which C. diff is a chronic diarrhea problem. Um, Another survey looking at what happens in relation to cannabis usage. There were two big surveys looking at cannabis users versus non-unit users, and obesity rates were distinctly lower, and people use cannabis even moderately. There was a one-third reduction. This wasn't related to tobacco smoking. So overall, it was felt that the use of cannabis was associated with body mass index differences in both of the surveys. Based on animal work, DIO mice are subject to becoming fat, and particularly when they're given a high-fat diet. Um, it was shown that THC altered the balance of two types of bacteria in the gut, Firmicutes and Bacteroidetes. THC prevented the ratio increase or weight gain despite the high-fat diet. So the question comes up. One stereotype is the lazy slacker stoner. This guy is obviously obese and a little slovenly in his habits, versus the skinny hippie stereotype. So which is really true? Well, in fact, it is the skinny hippie. And for people who use cannabis more regularly, they're apt to be lean. It's not just because their income may not be as great. So, what are people to do to increase their endocannabinoid tone? Well, aerobic activity is one thing, but again, on the diet side, the probiotics are those fermented bacteria. They're beneficial on the gut lining. These can come from yogurt, kefir, fermented vegetables, etc. And then the feedstocks for them, the prebiotics, that can be found in burdock, marshmallow, slippery elm, etc. Or just from your food, particularly the Leucae, your leeks, garlic, and onions, more exotic things like burdock root, dandelion greens, etc. All this is important as well because there may be benefits of all this in helping to prevent Alzheimer's disease. There has been an association between the acne bacteria, Propionibacterium acnes, which has been found in the brain of Alzheimer's disease victims. And it may be that probiotics, prebiotics, and the cannabinoids exert protective effects. If you're interested in this, I suggest the article that can be found at ethanrusso.org at the library tab. And finally, let's talk about other plants that affect the endocannabinoid system. So these are ones that affect the CB1 receptor in different ways. Kava kava is a well-known anti-anxiety social drink in the South Pacific. This radula marginata is from a liverwort, but this is not going to be commercially viable. It's a rare plant and it only grows millimeters a year. There are various of the TRPV1 agonists, of course, cannabidiol, as we've mentioned, and chili peppers, but ginger and black pepper also work on this receptor. FA inhibitors are going to increase endocannabinoid tone, and the most famous there is going to be chocolate, which contains these sort of endocannabinoid-like molecules. So there may be wisdom to the idea that that chocolate increases mood. And then miscellaneous agents. These are all interesting. We see black pepper here again. And in fact, truffles make anandamide, even though the plant, this actually is making it black. It isn't using it in the way that animals do. And we'll close with this very interesting plant called Helichrysum umbraculagerarum. It is a daisy-like plant that grows in southern Africa. Back in 1979, it was noted that cannabigerolic acid and cannabigerol were found in trace amounts in this plant. With my colleagues, we tried to look into this. It was a very hard plant to obtain. We didn't find commercially viable amounts of that, but a subsequent study by Berman et al. Showed that this plant has the potential for making hundreds of distinct cannabinoids beyond the 150 that have been described from cannabis itself. And with that, I will close and I'll stop sharing and hopefully we can answer your questions.
Thanks so much, Ethan. That was a great talk. So we have a few questions. I was going to ask you to comment on non-cannabis phytocannabinoids, which you did at the end of your talk. So you beat me to that one. We have there was also a question that came in about developments regarding new cannabinoids, and I think you're covering the last couple slides kind of address that. Is there anything else you want to
Oh,
say about that?
oh, sure. Beyond CBG, we didn't really have time to talk about tetrahydrocannabivarin. But again, this is a neutral antagonist. This has shown in humans the ability to really beneficially affect parameters of the metabolic syndrome and help people to lose weight in a way that will be cheaper and maybe safer than the GLP-1 drugs that everybody in Hollywood is taking. But I want to mention another one, well, a couple. People will hear a lot about delta 8 THC. Delta 8 is actually a natural component of cannabis, but in trace amounts. It is not something you could selectively breed for, but it's all over the landscape because there's been a glut of CBD. People have turned it synthetically into delta 8 THC, which is similar to delta 9 THC in its effects, The problem is not with the molecule delta 8 THC. Rather, it's how it's made and the fact that there's no quality control in the industry. What's advertised may not be what's in it. And virtually all of the delta 8 products that have been tested are contaminated with other cannabinoids that we know even less about, and often with residual solvents. So, warning there. Somewhat analogously, and a few years ago in Italy, they described something called tetrahydrocannabidiol, THCP. So this has a 7 carbon side chain instead of 5. It is 33 times more potent than THC. Nobody needs THCP. But again, it's unlikely that you'd ever selectively breed for this in amounts that would be significant. But there are THCP products that are available on the internet right now. And a lot of people have had very bad experiences with these. Keep in mind, THC is a weak partial agonist. It's working on a system that needs gentle nudges, not big pushes. And THCP is definitely a big push and a dangerous chemical the way it's currently available.
Thank you. All right, so I, I have my own question, which I like to ask, which is, um, you know, in some of your slides, you showed like the pre and post synaptic region of neurons, and so that prompted me to think about the interaction and relationship between the ECS and the general second messenger system. So, can you comment about that and how that might impact drug interactions?
Yeah. Okay. Well, some of this is beyond my pay grade. We really need a genuine
Uh,
pharmacologist. But what Reggie's talking about is when the compound hits the receptor, that's not the end of the story. There are all these intermediary steps, what's called trafficking. That's not like trafficking of drugs. It means whether the drug on that receptor is going to stimulate cyclic AMP or uh, act as a brake on cyclic AMP, and this will affect downstream effects like whether it interferes with certain aspects of cancer metabolism. Um, so yeah, that's the real granular detail that where I defer to my basic science colleagues. Um, but I hope people appreciate that this is an area of science that's still in development. It's extremely complex, and we need to resist the temptation to try and present things in an overly simplistic fashion. I've been studying this intensively for 30 years and still learn something new every day. So
so some of the other questions that came in, not sure whether or not you would have an opinion or be able to answer. One of them was on the effects of remediation and or quick dry processes and how that affects the flower in curing.
Uh, well, all right. Remediation. Remediation to me means that you've got a potentially contaminated product that you're trying to fix. This would most commonly be due to a couple of different things. One would be pesticides. The other would be polyaromatic hydrocarbons from drying with heat, like with propane dryers. My opinion is garbage in, garbage out. If you got something that's contaminated, that should be end of story. Discard, try again, do it right. There are techniques available to take that material and extract it and ideally get rid of the bad stuff. But that's just encouraging bad technique. It's like the idea of uh, using Roundup to grow plants so that you can indiscriminately spray herbicides on a field. Bad policy, in my opinion. The second part was about drying. I would question why we dry. Drying is done to produce a better smoke. However, it neglects the fact that most cannabis products, especially ones used medically, shouldn't be smoked or would be available in another form. Drying invariably is going to lead to loss, particularly of the terpenoids. And if it's not done properly, it can easily be half the terpenoids will be gone. Um, there are slow processes done at a low temperature with regulated humidity over a couple of weeks that can mitigate that. But drying is, again, primarily for the recreational market of cannabis flower and may not be necessary at all for other extraction techniques, particularly for the medical markets.
Let's see here. We only have like one or two more. So one just came in that is asking about. I am curious about your thought on naturally occurring isomers of THC and enantiomers, diastereomers, and different ratios reported in hemp and THC dominant chemovars. Um, the two THC plants may harbor slight differences in pharmacology. What about the role of isomers in pharmacology? Thanks for a great presentation.
Um, yeah, what we're talking about is the three-dimensional structure. So in plants, often the same molecule with the same molecular weight will come in two different flavors. And one version might work on the receptor and the other one may not. Um, uh, this really hasn't been worked out thoroughly. Um, and it becomes a question of should we be dealing with one isomer, one particular three-dimensional structure is preferable to others. Um, for the most part, the consumer is not going to be able to tell the difference. Um, once it were the case that we have a change in the laws and the better ability to research these kinds of questions, we could answer these questions more effectively. So that's sort of a dodge. But suffice it to say that these differences exist. How that is important on a practical level, I really can't say at this time.
Okay. And the last question we're going to address right now, and I can answer this one, Ethan, is so somebody asked, how have new farms been allowed to replace the low quality government grown cannabis? The answer to that is yes. Okay. A couple years back, the DEA actually issued licenses to, I think it was five or six additional DEA Schedule 1 cannabis producers. They are located around the United States. There are some, actually, another fairly well-known cannabis researcher, Sue Sisley, actually has one of those licenses. So do you want to add anything to that, Ethan?
Yeah, as uh, at this time, I'm not sure how much those are being utilized. There are places like the University of Colorado that have become very adept at finding workarounds. So they have a van that they will take out, let people smoke their preferred cannabis, come into the van, get tested in whatever fashion, etc. Heretofore, it's been really difficult to do this kind of research because in most instances, the FDA has required what's called an investigational new drug application, which is a bureaucratic nightmare for the average researcher in terms of time and expense. But it has become more possible to study the kind of cannabis that is available to consumers from dispensaries. Um, in each instance, I would advocate that we need to know its composition. My preference is that any such study that's published should be accompanied by a certificate of analysis. It's the only way to know if we're doing apples to apples comparisons. Um, so again, because there's so many different components in cannabis, it is probably preferable to avoid generalizations like if this one worked, then that one will work. It only works when it's proven to work.
Okay. I actually missed one question that I found pretty interesting. I have my own opinion on this, but the question is, will there ever be a way to accurately determine cannabis intoxication while driving?
Well, I'll let you go first and then I'll follow on if necessary.
So my opinion is uh, because of the way. So let me start a different way. So a lot of the methodology that people have adopted or tried to put into place is based on the alcohol model, which is completely different than the cannabis model, right? So alcohol has a very specific interaction with the body. It's a dehydration reaction. So because of that, there is a fairly constant body mass to percent alcohol ratio that you can look at that will give you that intoxicated reaction. That is not the case for cannabis because of the differences in people's genetics, the number of CB1, CB2 receptors, that kind of thing. Everybody's got a different amount of fat on them. The cannabinoids are very fat soluble. And so in my opinion, I do not believe that there will be a breathalyzer type of um, approach that will be successful. Now, whether or not you can use a behavior modification type of test where you look for delay in response or potentially um, uh, memory or some other type of approach. That's yet to be seen, but those are going to be very different and will likely not be roadside kind of approaches. So that's my opinion on that. So
Right. So yeah, I agree. Just to expand, I agree wholeheartedly. There's no blood test, saliva test, hair test that's going to indicate impairment. Rather, this has got to be done behaviorally. So trained examiners can measure impairment from cannabis potentially with driving. But that requires training. You're not going to have someone who's trained in every squad car that makes a roadside stop. But that's the only reasonable measure because there's no correlation between serum THC levels and impairment. It's in what's in the brain that counts, and we don't have roadside scans for that. So it's a very sticky problem.
um, and I'm just checking to make sure that I've gotten everything. Um, there were some questions regarding additional infrastructure needed to carry out cannabis research. I will deal with those uh, directly by email because that's going to be, I think, a little bit more complicated than we can deal with here. Um, and that's it. Let me
Oh,
just.
well, Reggie, I know we have just 10 more minutes about, and there's some in the Q&A that I would love to highlight, and I've also put in.
Oh, okay. So one did just
Oh,
come in. Sorry about that.
yeah,
Um,
fair to ask.
Mhm.
let's see here. Nate, where does Dr. Russo see? Oh, sorry about that. Where does Dr. Russo see this sort of research going next? What are the next questions and gaps that need to be filled?
Well, tons. I mean, we've hit on some, but what's most exciting to me is the minor cannabinoids. So again, we've tried to spearhead research on cannabigerol, which is proving quite promising. But THCV, some of the variants, the propylcannabinoids, every phytocannabinoid that's been examined has weird and wonderful properties of its own from a therapeutic standpoint. So there's plenty left to do. Following from that, selective breeding for these particular agents. Reggie knows plenty about that. So yeah, the future is bright if we can do this kind of work. But right now, funding is a huge problem, and legal constraints remain a huge problem.
Since THCV is an antagonist of CB1, do we have any indication on how THCV works for reducing epileptic seizures?
Uh, that one has been controversial. There have been some studies that show an anticonvulsant effect, and others that it's been less clear. So there have been no clinical trials of it. However, in medical applications, often more is better in terms of the diversity of different cannabinoids. And it's not only a matter of efficacy, it is a matter of tolerability. My strong feeling is that these added agents really temper the effects of THC and make it a more acceptable experience, lowering that adverse event profile. The nitty gritty of it, a lot of these things may be working at a mitochondrial level, affecting energy balance. And for example, some of the rarer syndromes that produce epilepsy are mitochondrial diseases. So there's plenty of grist for the mill there as well.
Let's see here. In the myths on your slide, it said CBD can be psychoactive. Can you expand and what amounts? Yeah. So
Okay, so again, psychoactive means that it affects activity in the brain, specifically on anxiety. We know that in testing of social anxiety, Bergamaschi et al. In Brazil showed that 600 milligrams could notably reduce social anxiety on testing in humans. So that's a big, big dose, not practical for anybody in terms of expense. Similarly, there have been 2 phase 2 clinical trials looking at CBD as an antipsychotic agent to treat schizophrenic patients. What they showed was that at 800 to 1,000 milligrams of pure CBD a day, that these were equivalent in efficacy to standard antipsychotic drugs and with fewer side effects. So those have to be considered psychoactive. Again, we make a distinction from intoxication.
Piggybacking on that, a recent paper came out. I forget the author that suggested. Actually, it was in the last couple of weeks that suggested that the use of CBD increased enzyme levels in the liver. So, can you comment on?
Right.
On whether or not that is a general effect or is that specific to a certain subset of the population? Because
Yeah,
my
I'm
understanding
really
is that
glad
it's
you asked that. Sure. So, yes, CBD will compete for cytochrome P450 enzyme, and drug-drug interactions are the result. However, this has been closely studied, and you only see those drug-drug interactions with CBD in doses above 300 milligrams a day. The most common context is with Epidiolex, very, very high doses, 1,500 milligrams or more a day, in conjunction with polypharmacy. So usually, and most often, these are children, they're on multiple other anticonvulsants. So there are definite drug-drug interactions that are possible with the anticonvulsants, particularly with clobazam, that combination will increase the amount of N-dysmethylclobazam, a sedating breakdown product of, it's a metabolite of clobazam. Also, a lot of drug-drug interactions with valproic acid, but valproic acid is a bad actor. It doesn't play well with others and is best used as a sole agent, not in combination. So again, what I'm going to say as a blanket statement is we don't need to worry much about drug-drug interactions or, the liver enzyme elevation, again, is usually in polypharmacy with extreme doses. There's nothing to indicate that lower doses of CBD, particularly in extracts rather than as an isolate, are going to produce hepatic enzyme elevations.
Um, it looks like so there's one more question, which is, can you talk a little more on the interaction between serotonin system and the ECS?
Oh, um,
If
complicated.
it,
There are,
if that's too much for right now, we can
well,
just say that. So,
let's try and be a little more specific. There are 30 different serotonin receptor subtypes, and how they act is extraordinarily complex because they can be in multiple different parts of the neuronal assemblage. What we know is that CBD specifically acts as a stimulator for the serotonin 1A receptor. This helps explain how it is an anti-anxiety agent, but it also has a role in these other disorders, damage after stroke, vomiting, nausea and vomiting, etc. But boy, that's worth a whole semester course on its own.
all right.
Somebody's
Uh,
interested in that, I'd encourage him to pursue it as a graduate thesis.
Okay, so for any of you still here that are listening, if you guys want to have a very robust graduate thesis project, there you go. You heard it from Ethan himself. So, and on that note, I'm going to say thank you for joining us, everyone. Ethan, thank you so much for kicking this off for us. I really appreciate it. I'm a big fan of yours. I have been for a while. I am honored that you gave us the time today. We will be making uh, uh, this presentation available. We've got it recorded. We will. I don't know where we're going to post it yet, but we will post it somewhere. And I will. You can reach out to us. Through that QR code, or we have we don't have our info at kind of email address here, but either way, reach out to us at CRI either through LinkedIn or whatever. We will, and we'll share it in the link in our next newsletter as well. And stay connected with us. Subscribe to our newsletter. Visit our website. Follow us on LinkedIn and connect with us directly. Once again, thanks, Ethan, so much for the time.
Thank you. Cheers.